In 1995, Australia was facing an agricultural crisis that most people have completely forgotten about. Rabbits had overrun the entire continent. They were eating crops, destroying grazing land, and costing farmers millions of dollars every single year. The government had tried everything to stop them. They'd spent decades and millions on poison, fences, and hunting campaigns. Nothing worked. The rabbits kept reproducing faster than anything could kill them. Then, in 1995, something changed. A disease escaped from a research facility. Within weeks, it killed 99% of the rabbit population across the entire country. The rabbit plague was over. But what happened at that facility, what the government was actually testing, and how one escaped virus became the most successful biological control program in human history, is a story about desperation, science that went wrong, and the way sometimes our solutions create new problems we never saw coming. And before we go further, I need to pause for a second. If you're watching this because you're worried about feeding your family, about making ends meet, or about how you're going to pay for food this week, I want you to know you're not alone. That's why I want to tell you about a resource that literally changed the way people approach their grocery bills. There's a platform where people share exact, down-to-the-dollar meal plans, bulk buying strategies, and how to stretch $50 into a week's worth of meals for a family of 4. It's not a diet program. It's not affiliate marketing. It's actual people writing down what they buy, where they buy it, and what it costs. Some of those strategies are going to save you money this month. Now, let's go back to 1995. The story of Australia's rabbit problem starts more than a century earlier, in the 1850s. A man named Thomas Austin, a wealthy landowner in Victoria, decided to import rabbits from England. He brought a few dozen with him because he wanted something familiar. He wanted to hunt rabbits like he did back home. He wanted to relive the English countryside in the Australian outback. He didn't think much about what would happen if those rabbits escaped. He didn't spend time worrying about the ecological consequences of introducing a non-native species to an isolated continent. He was a businessman, a landowner, someone used to getting what he wanted and making decisions that affected large areas of land. He released the rabbits onto his property. He thought they would stay there. They did. And when they did, something catastrophic unfolded. Rabbits had no natural predators in Australia. There were no foxes, no badgers, no hawks that had learned to hunt them. The climate was perfect for rabbit reproduction. The grasslands were unlimited. They had more food than they could eat. They had more space than they could possibly need. The population explosion that followed was one of the most dramatic biological expansions in recorded history. Within 50 years, there were millions of them. Within 100 years, there were billions. A rabbit plague had consumed an entire continent. The spread wasn't gradual, it was exponential. Rabbits can breed year-round in warm climates. A single doe can have up to 5 litters per year. Each litter can contain 10 or more kits. Do the math. 100 rabbits becomes 1,000, becomes 10,000, becomes 100,000. By the time the government realized there was a problem, the problem had already conquered the country. By the 1950s, the spread of rabbits across Australia was one of the fastest biological invasions in recorded history. Scientists studying invasive species used the Australian rabbit expansion as a model case. It was the textbook example of what happens when you introduce a generalist species with no predators into a permissive environment. The rabbits had reached the ocean on both coasts. They were everywhere. They were in the deserts, moving across the red earth like a plague out of the Old Testament. They were in the forests, eating the understory vegetation. They were in the grasslands that farmers depended on for their livestock. They were in the agricultural districts that should have been feeding the nation. The damage was staggering. A single rabbit eats about half a pound of vegetation every day. That doesn't sound like much. But when you multiply that by 1 billion rabbits, you get about 500 million pounds of vegetation disappearing every single day. Every single day. That was vegetation that could have fed sheep, that could have fed cattle, that could have grown into crops. That was grass that was supposed to be grazing land. That was understory vegetation that was supposed to support native wildlife. The economic impact on farming communities was devastating. Farmers were watching their land being consumed. They were watching their animals starve because there was no grass left. They were watching their income disappear, eaten by rabbits. Farmers couldn't compete with the rabbits. They tried hunting them. Men with rifles would go out into the countryside and shoot rabbits for days. They would come back with dozens, hundreds. It would barely make a dent in the population. The rabbits would breed back the losses in weeks. They tried poison. They used 1,080. They used strychnine. They used every poison they could think of. The rabbits died. But the ones that survived, the ones with slightly different digestive systems or slightly different metabolism, they passed that resistance to their offspring. Within a few generations, the poison became less effective. The rabbits adapted. They tried fencing. Farmers built rabbit-proof fences, long barriers meant to keep the rabbits out of protected areas. The rabbits dug under them. They dug with their powerful front legs. They moved tons of dirt. They found every weakness in every fence. They got through. They tried introducing predators. They brought in animals from other places. They hoped that natural predators would control the rabbit population. That didn't work either. The predators were outnumbered. The predators couldn't breed fast enough to keep up with the rabbit reproduction. The predators sometimes focused on native species that were less abundant than rabbits. Nothing worked. Nothing seemed to work. By the time the 1980s arrived, Australia had tried everything Western science could think of. They'd spent millions on Myxomatosis, a disease that had been introduced to Australia in the 1950s with some success. The Myxomatosis worked for a while. It would spread through the rabbit population. It would kill a lot of rabbits. The problem would seem to be solved, but then the rabbits would develop immunity. The virus would mutate, the rabbits would mutate. An arms race developed between the virus and the rabbit population. The virus was still killing some rabbits, but it wasn't enough. The vaccine had lost its power. The population was still growing. The problem was still catastrophic. Scientists could see that Myxoma was no longer adequate. They needed something new. They needed something more powerful. And then, in the late 1980s, the Australian government made a decision that would change everything. They decided to fund a secret research program. The program was run by the CSIRO, Australia's main scientific research agency. The CSIRO was the country's primary center for scientific research. It had access to funding. It had access to equipment. It had access to the brightest scientists in the country. The goal was simple but massive. They wanted to find a new biological weapon against rabbits. Not a poison that the rabbits could adapt to. Not a virus that would lose effectiveness over time the way Myxoma had. They wanted something new, something better, something that would work permanently or at least for decades, something that would finally end the rabbit plague. They wanted a silver bullet, a biological solution so powerful that it would reshape the entire continent. The researchers started working with a virus called rabbit hemorrhagic disease. It wasn't discovered in Australia. It came from Europe, from China, maybe even further back. Nobody was completely sure about the origins. The virus had appeared in wild rabbit populations in Europe in the 1950s. Chinese researchers had documented it in domestic rabbit farms in the 1980s. It was moving around the world through trade routes and wild populations. The virus had been studied quietly by scientists in several countries. What they knew was that the virus was deadly to rabbits. It was called hemorrhagic disease because that's what happened to infected animals. It attacked the liver. It caused massive internal bleeding. Blood would appear in the lungs, in the kidneys, throughout the abdomen. It killed rabbits in days, sometimes in hours. The fatality rate was almost 100%. When a rabbit got infected, it had a very short time left. It might survive for 24 hours. It might die in 12. There was almost no recovery. The researchers at CSIRO understood immediately what they had. This was the solution they'd been looking for. This was the biological weapon that could solve Australia's rabbit problem. They started working with the virus in contained laboratory conditions. They kept samples in a laboratory that was supposed to be secure, supposed to be safe. They studied how it spread. They studied how quickly it killed. They studied the mechanisms of infection. They studied which populations of rabbits were most susceptible. They ran experiments to see if they could use it as a biological control agent. They wanted to understand it completely before they released it. The idea was simple: release the virus into the wild rabbit population. Let it spread naturally. Let it kill the rabbits on its own timeline. Solve the problem without any government intervention once the virus was out there. But biological weapons are dangerous. The researchers knew that. The government knew that. That's why the work was classified. That's why most of the public didn't even know what was happening in those labs. The facility where the research was being conducted was kept secret. The details of the research were kept secret. The sample collections were kept secret. This was the kind of research that nobody talked about. In the early 1990s, something went wrong. There are several versions of how it happened. Some official reports say it was an accident during transport. A shipment of infected research rabbits or contaminated material was being moved from one facility to another. Someone didn't follow proper protocols. Someone didn't use proper containment. The material escaped. Some reports say it was a deliberate leak. Someone inside the facility, someone who understood the urgency of the rabbit problem, someone who was frustrated by bureaucratic delays, deliberately released the virus, someone who decided that the only way to solve the problem fast enough was to let it loose. What we know for certain is that in 1995, rabbit hemorrhagic disease escaped from the CSIRO facility. The virus got out of containment. It infected wild rabbits around the research area. And then, something remarkable happened. The disease spread across the entire continent in weeks. Not months, not a season, weeks. It moved across grasslands and deserts like nothing anyone had ever seen. It moved through forests and farmland. It moved through remote areas that had no human population. It simply moved. Scientists couldn't track how fast it was moving, because it was moving faster than they could measure it. They would go out to collect samples at a location they'd gotten reports from. By the time they got back to the lab, the outbreak had moved hundreds of miles. The virus was racing across Australia like nothing they'd ever seen before. It was moving like it had been designed to move. It was moving like a weapon. The first confirmed death of a wild rabbit from rabbit hemorrhagic disease in Australia was reported in June 1995 in Victoria. Within 1 month, the virus was detected in New South Wales. The news came from farmers. They started calling authorities. They started reporting dead rabbits, rabbits that were behaving strangely, rabbits that were dying in their burrows. Within 2 months, the virus was in Queensland and South Australia. The spread was now undeniable. It was moving in multiple directions simultaneously. Within 3 months, it had reached Western Australia. It had crossed the entire continent. The speed of the spread was incomprehensible. It was moving faster than migratory birds. It was moving faster than any natural vector could explain. It was moving like it had been designed to move across a continent exactly this size. And as the virus spread, the rabbit population began to die. Not gradually, not slowly, rapidly, massively, catastrophically. It was as if the entire rabbit population was being erased from the landscape. Dead rabbits appeared in massive numbers. Whole warrens died simultaneously. Entire ecosystems where rabbits had been the dominant herbivore suddenly went quiet. The ecological change was visible. Farmers who had spent decades fighting rabbits, who had lost crops and income to rabbits, who had almost given up hope, started noticing something miraculous. The rabbits were gone. The fields were empty. The grasslands were recovering. The grass that had been eaten to the ground was starting to grow back. The disease was working. The biological weapon was working exactly as planned or as accidentally desired. But the death rate was almost incomprehensible. Scientists estimated that in the first few months after the escape, the rabbit hemorrhagic disease killed about 95% of Australia's wild rabbit population. Some estimates put it even higher. In some regions, the kill rate was as high as 99%. In Western Australia, the kill rate appeared to be nearly 100%. The continent that had been overrun by rabbits for 150 years was suddenly, almost overnight, almost rabbit-free. Not completely rabbit-free. Some rabbits survived. Some rabbits had genetic resistance. Some rabbits were isolated in warrens that the virus couldn't reach. Some rabbits survived in pockets of the population that were protected by geography or by chance, but the overwhelming majority of the rabbits, the billions of rabbits that had consumed the continent, were dead. The ecological impact was immediate and dramatic. The landscape started to recover in real time. The vegetation that rabbits had been eating for generations began to grow back. Native plants that hadn't been able to establish themselves started to recolonize areas. The grass species that make up the Australian savanna began to recover. The understory vegetation in forests started to come back. Predators that hadn't been able to hunt rabbits successfully switched to other prey. Feral foxes that had been focusing on rabbits switched to smaller marsupials. Raptors changed their hunting patterns. The entire ecosystem began to rebalance itself. The change was visible from satellite. Researchers could see it in satellite imagery. The green of vegetation was coming back. The land that had looked like a bare, grazed desert was starting to look green again. The entire continent was healing. The question everyone asked was simple. How did this happen? How did a virus escape from a laboratory and spread so completely, so quickly, across an entire continent? The official answer from the government was always vague. They said it was an accident. They said the containment had failed. They promised it would never happen again. But the truth was more complicated. Rabbit hemorrhagic disease is spread by insects, particularly rabbits, fleas, and mosquitoes. It can also be spread through direct contact with infected rabbits. The virus spreads faster in warm weather. It can survive on surfaces. It can travel through water. Once it got out of the lab, all of those vectors of transmission were working together to spread it as fast as possible. Summer in Australia is warm. It's humid. It's perfect for a virus that spreads through insects. The timing of the escape, if it wasn't deliberate, was almost too convenient. It happened right at the start of warm season. It happened right when the conditions were optimal for the fastest spread. Some people have suspected that the escape wasn't actually an accident. Some people have suggested that someone inside the research facility, someone who understood the urgency of the rabbit problem, deliberately let the virus out. Someone who decided that the government's bureaucratic process was too slow and that the only way to save Australia's farming industry was to release the virus and let it spread. If that's true, no one has ever admitted it. No official investigation ever proved it. But the suspicion has never gone away. The documents about the escape remain classified in many cases. The full details about what happened at the CSIRO facility in 1995 are still not public. What is clear is that the rabbit hemorrhagic disease escape was either the luckiest accident in Australian history or the most successful deliberate biological weapon release in human history. Either way, it worked. By the end of 1995, the rabbit problem that had plagued Australia for 150 years was essentially over. The economic benefit to Australian agriculture was enormous. Farmers suddenly had their grasslands back. Crops weren't being destroyed anymore. The cost of rabbit control dropped from millions of dollars a year to almost nothing. The money that had been spent on hunting, poison, and fencing could now be spent on other things. The farmers could focus on actually raising livestock and growing crops instead of fighting a never-ending war against rabbits. The ecological benefit was just as significant. Without rabbits eating everything in sight, the Australian landscape began to recover. Plants that hadn't been able to establish themselves for generations started to come back. Animals that had been squeezed out by the rabbit population had more resources to survive on. The biodiversity of Australia started to improve. The soil, which had been trampled and denuded by billions of rabbit feet, had a chance to regenerate. But the virus didn't stop at 99%. It didn't just kill the rabbits and then disappear. It stayed in the environment. It became endemic. It became part of the Australian ecosystem. Every time the rabbit population starts to recover, the virus kills them back down. In the years after 1995, the rabbit population would occasionally spike. Then the virus would spread again, and the population would crash again. It was like nature had created a thermostat for the rabbit population. Too many rabbits, the virus spreads. Fewer rabbits, the virus dies down. The system was in balance. But the virus was constantly mutating. Scientists noticed that some rabbits were developing resistance to it. Some strains of the virus were becoming less lethal. This is what happens with any biological control. It's not a permanent solution. It's a temporary suppression that has to be managed constantly. The government had to keep paying researchers to monitor the virus. They had to keep studying how it was changing. They had to be ready for the possibility that one day the virus might lose its effectiveness entirely, and the rabbit problem might come back. There's a dark irony in the story of the rabbit hemorrhagic disease escape. The Australian government had spent decades trying to solve the rabbit problem through every conventional means. They'd tried poisons that the rabbits adapted to. They'd tried hunting campaigns that the rabbits outpaced. They'd tried introducing predators that the rabbits somehow survived. They'd spent millions and millions of dollars, and nothing had worked. Then, a virus escaped from a laboratory, and it solved the problem in weeks. The problem that was supposed to be unsolvable was solved by an accident. Or by something that might have been an accident. The lesson was uncomfortable. The government had to admit that the only way to really control the rabbit population was through biological force that they couldn't fully contain or control. The only way to win was to release something they didn't fully understand and hope that it would do what they wanted it to do. That's not the kind of solution that gives you confidence in government control of dangerous things. The rabbit hemorrhagic disease escape also raised serious questions about biosecurity that are still relevant today. A virus that could kill an entire species escaped from a supposedly secure laboratory. If it had been a human pathogen, if it had been designed to attack humans instead of rabbits, the consequences would have been unimaginable. The escape showed that laboratory containment isn't perfect. The most careful protocols can still fail. Someone can still make a mistake, or someone can still decide that breaking the rules is necessary. The facility was upgraded after the escape. New safety procedures were implemented. New monitoring systems were put in place. But the basic vulnerability remains. A virus can escape. A disease can spread. A weapon can get loose. The question of intentionality has never been resolved. This is the part of the story that matters. This is the part that haunts the entire episode. The Australian government maintains that it was an accident. They say that a shipment of infected rabbits or contaminated materials accidentally escaped during transport. They say that someone didn't follow proper procedures. They say that nobody deliberately released the virus. This is the official story. But the timing was too perfect. The spread was too fast. The success was too complete. The virus didn't escape in winter when it wouldn't spread as fast. It escaped in spring, when the weather was warming up, when insects were becoming active, when the conditions for viral spread were optimal. The virus spread east and west simultaneously. If it had escaped from a single laboratory, why did it spread in multiple directions at once? Why did it appear in remote areas that had no connection to any research facility? A lot of people have always suspected that someone made a decision, someone inside the system, someone who understood the urgency of the rabbit problem, someone who was frustrated by the slow pace of government bureaucracy, someone who decided that things had to change, someone decided that the rabbit problem was urgent enough to justify releasing a biological agent without going through all the bureaucratic steps. Someone decided that the rules didn't apply when the stakes were this high. Someone decided that the ends justified the means. That person was never identified. That person was never punished. That person was never even officially named. The government never conducted a serious investigation into who released the virus. They never publicly investigated how it escaped. They released official statements about accident and containment failure, but they didn't explain how a contained virus ended up in multiple locations simultaneously. They didn't explain how it spread so fast that scientists couldn't track it. If someone inside that laboratory deliberately released the virus, they are probably still alive today. They might be retired. They might be living somewhere in Australia or somewhere else entirely. They might read news stories about how the rabbit plague was solved and smile to themselves, knowing that they had something to do with it. They knew something that nobody else was supposed to know. They broke the rules. They released a biological weapon on their own country, and it worked. That person, if they existed, probably saved Australia billions of dollars in agricultural losses. That person, if they existed, also set a precedent that nobody wants to talk about. They showed that the rules could be broken if you had a good enough reason. They showed that biological weapons could be used domestically on your own country's land for what could be justified as the greater good. They showed that if you wanted something badly enough, you could act unilaterally. You could break the protocols. You could risk the consequences. And if you were right, if the consequences turned out to be beneficial, you would never be punished. You would probably never even be caught. They opened a door that, in a more dangerous world, might have led somewhere very bad, somewhere even worse than Australia's rabbit plague. What if the next person who faced a problem thought they could use biological agents? What if the next problem wasn't rabbits? What if the next person who broke the rules was trying to solve something they didn't understand? What if the next escape was actual biological weapon instead of just a rabbit virus? The rabbit hemorrhagic disease escape is sometimes taught in textbooks as a success story. The story of how biological control saved Australia. The story of how science solved an impossible problem. The story of how a desperate situation got resolved. But it's also a cautionary tale about the dangers of uncontrolled experiments. It's a story about the way that solutions can escape and create new problems. It's a story about the fine line between biological control and biological weapons. It's a story about the way that the most important decisions are sometimes made by accident or by people who break the rules in the middle of the night, and nobody ever really knows for sure what happened. By the year 2000, rabbit populations had stabilized at about 1% of their pre-1995 levels. The ecological recovery was remarkable. Areas that had been completely denuded by rabbits were coming back. Vegetation was returning. Animals were recovering. The agricultural benefit was measured in billions of dollars. Australia's farming industry was saved. The threat that had seemed permanent was gone. The problem that had seemed impossible to solve was solved. But the virus is still out there. It's still circulating in the rabbit population. It's still maintaining the population at low levels. Scientists are still monitoring it. Researchers are still studying it. Because the day that the virus loses effectiveness, the day that the rabbit population starts to adapt and develop immunity, is the day that Australia might have to face the whole problem all over again. The virus can't be recalled. The genie can't be put back in the bottle. Once a biological agent is released into the environment, it's part of that environment forever. It mutates. It spreads. It evolves. It becomes something that nobody can fully control or predict. That's the lesson that Australia learned in 1995. Whether the escape was an accident or not, that's the lesson that the world should have learned. That's the lesson that seems to get forgotten every time we think we have a problem that only a biological solution can solve. The story of the rabbit plague and the virus that ended it is a story about what happens when nature is pushed too hard, when solutions are pushed harder, and when the desperate need to solve a problem overrides the careful protocols designed to keep dangerous things contained. It's a story about a continent that was almost destroyed by rabbits and then saved by a virus. It's a story about the way that one decision, one accident, one escape can reshape an entire ecosystem and an entire industry and an entire nation's approach to the problems that seem impossible to solve. Australia's rabbit problem is gone now, but the virus remains. It remains as a reminder. It remains as a warning. It remains as proof that sometimes the solutions we create are almost as dangerous as the problems they're meant to solve.